This call is now being recorded.
So, our local chicken crosses in our backyard with exotic chicken. The crossbred, the F1, will have superior advantage over the two parents. The crossbred will be bigger than the local chicken, and the crossbred will have disease-resistant advantage than the exotic chicken. Heterosis will be in action. Hybrid figure will manifest it. That is crossbreeding, manipulation of genes. But the traditional genetic engineering is just mere crossbreeding. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. It's just genetic, it's just crossbreeding. Example, another example is when you know what they call the mule. Mule is generated from traditional genetic engineering. Traditional genetic engineering, which is crossbreeding. When you cross a stallion male with a jenny, a female donkey, and you have male. We are trying to transfer the strength of the horse onto the gentle donkey. We want to increase the strength of donkey so that those may have more strength. So we have mule, which is F1, which manifested heterosis and hybrid figure. The mule is bigger than the parent donkey and has more strength, than the female, than the parent donkey. We have mule, but advantage is mule is sterile because of chromosome differential. For animals to be able to interbreed, they must share the same number of chromosomes. If they don't share the same number of chromosomes, you may succeed in crossing them, but they may not succeed in reproducing. In fact, in some cases, due to differential chromosome number, they will not even, there will not be fertilization. It's the first place. So mule is a form of traditional genetic engineering, where you have mule that has strength of the horse, that have the calmness of donkey, and at the same time, the strength of the horse. Another one is we have Jenny. Oh no, we have Zendok. When you know zebra is beautiful, and donkey is not really beautiful. Donkey and donkey is not. Okay, take for instance the bull that was produced as a result of crossing between stallion, a male horse, and the jenny, a female donkey. The stallion will transfer strength and big body side onto a donkey, and the F1 will be bigger than the female, than the parent donkey. Much stronger than the parent donkey. Now, the same thing in Zendok. When you cross male zebra with jenny, female donkey, you have Zendok. Though they cannot reproduce, they cannot reproduce. But we can be using F1 to be generating them. Crossbreeding to be generating them. We can be using F1 to be generating them. So anytime we hit them, we generate them. Now, this Zendok will have the beauty color of zebra and the body size of zebra, and the strength and the calmness or the resilience. Donkey has resilience. That's resilience. So the Zendok or the mule, you are able to fuse. Let me say Zendok, for instance. You have brought in the gene for beautiful color, gene for big body size into the Zendok, and the resilience of donkey into the Zendok. So Zendok will have performance superior and over and above the performance of the two parents. You see heterosis in action. You see a hybrid vigor in action. In genetic engineering, But traditional genetic engineering, I may ask you, with the help of empirical evidence or empirical demonstration, differentiate between traditional genetic engineering and modern genetic engineering. Traditional genetic engineering involve mere crossbreeding, maybe crossbreeding or hybriding techniques, like crossbreeding or strain crossing, crossbreeding or strain crossing, or hybridization of species, or line crossing, or pop crossing, or grading up. All those outbreeding techniques method can be used to produce, to genetically manipulate. Genetic engineering is gene manipulation, is genetic manipulation. It's not really gene editing per se. Gene editing is quite different. We are not removing any gene. We are just adding a gene. Okay, because when you add, we're not removing gene, but we are adding gene. That is gene manipulation. That is genetic engineering. Though gene editing is a form of it's also a form of genetic engineering. Like when you use CRISPR. When we use CRISPR, we can use it to cut certain segment of DNA and remove it. Possibly that is deletion. We use CRISPR to cause deletion. If that segment is causing harm, and let us remove this segment from the sequence of the DNA. So gene editing can either be removal or addition. So gene editing is gene manipulation. It's genetic manipulation. So there is more than molecular tools, molecular techniques, genetic engineering techniques. Because genetic engineering produces transgenic organisms, especially when we are manipulating genes. We are bringing two genes together in an individual. So meal, which was produced through traditional genetic hybridization, example of hybridization method. The meal is transgenic. Zendoc is transgenic. Another one is zebroid. When you cross, you want to increase the beauty of us. And we now cross zebra with horse, with male horse. You have zebroid. It is horse, but having beautiful colors of zebra. These are traditional transgenic, these are traditional genetic engineering. And these are transgenic organisms. Because we have transferred gene from one organism into another one. It's transgenic organisms. The organism carrying foreign DNA from two individuals. Transgenic organisms. So genetic engineering can be done. We have modern genetic engineering and traditional genetic engineering. Now, let us speak to modern genetic engineering, which is now prominent. Am I speaking sense? Am I speaking sense? Fantastic. This network disappears and okay. Now we have seen the tools to do traditional genetic engineering. Those are breeding matrix systems, which I've explained. Now, when we speak to let's speak to modern genetic engineering, which involve modern use of molecular tools. Modern use of molecular tools. So now we use traditional in traditional genetic engineering where we use breeding system, outbreeding system. Now we use transgenic techniques in generating transgenic organisms in modern genetic engineering. In modern genetic engineering, we are using now. See, in traditional genetic engineering, it is an organismal breeding, organismal exchange of gene. While traditional genetic engineering can be regarded as organismal genetic engineering. Why modern genetic engineering can be regarded as genetic engineering using modern molecular tools. And we have a wide range of transgenic techniques that is being used in genetic engineering. Gene transfer. In traditional genetic engineering, we use organismal method of gene transfer, where we use crossbreeding or outbreeding methods as a system at organismal level. All those crossbreeding methods or outbreeding methods like crossbreeding, strain crossing, grading up, top crossing, species hybridization are mating systems. Example of outbreeding mating system, which are gene transfer method in traditional genetic engineering. But for modern genetic engineering, to produce transgenic organisms, which are called GMOs, but GMOs are transgenic organisms because we use one gene to improve another. Am I speaking sense? And we have seen this virtually in traditional genetic engineering. Am I speaking sense? 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 Fantastic. Now, let me broaden your understanding about traditional genetic engineering. For instance, in environment, you cross. Let me say we have a local breed that are not being selected for milk production. But Chokoto Dugali is dual purpose. Chokoto Budali breed of cattle can produce milk and can produce beef. So we can use Fresian or New Jersey to improve using crossbreeding methods to improve our local breed, to improve the milk quality and milk quantity. That is crossbreeding method here is a form of gene transfer to create an F1 that will be able to produce higher milk yield, thrive better in our environment. Just will do, and those will do. So the Jesse cross will have superior performance. These are genetic engineering. These are gene manipulation, but the traditional way. So another thing is this. I told you genetic engineering is a gene manipulation. For instance, environmental biotechnology. You know, in biotechnology, we have animal biotechnology, we have environmental biotechnology, we have agricultural biotechnology, we have food biotechnology. In environmental biotechnology, in the era of oil spillage, there are organisms that feed on hydrocarbons. So research found that some organisms feed on hydrocarbons. So such organisms thrive where in hydrocarbon in impacted environment. So Since they feed on hydrocarbons, we can't use them to clean an hydrocarbon polluted environment. Do so because they have catechol gene in them. Catechol gene, the hydrogenase. This catechol gene confers the ability to be able to feed on hydrocarbons by not doing the cleaning. That is mutualistic. They are feeding on that mutualistic relationship. They are feeding on hydrocarbon, they are cleaning the environment for us. So we can isolate from these hydrocarbonoclassic bacteria. Hydrocarbonoclassic bacteria feed on hydrocarbons. So, but we have a whole lot of bacteria that don't have catechol gene. Catechol gene makes this conferability on these hydrocarbonoclassic bacteria to feed on hydrocarbons. So we can isolate this catechol gene, integrate it into other bacteria. So the gene will confer the ability to clean bacteria, to clean environment. So when we have oil spillage, just produce bacteria, transgenic bacteria carrying catechol gene, and introduce to the oil polluted area. They begin to clean it. Genetic engineering. Isn't that good? Isn't that making benefit for man? Of course it is. So that area of environmental biotechnology. Let's go to food biotechnology. The production of palm wine is biotechnology. So we use enzymes like invertase, am I correct? Invertase and all of that. Fermentation process. Fermentation process is traditional biotechnology. It's traditional genetic engineering where we use organism to ferment. We introduce organism to ferment our palm wine. So if you already see a fermented palm wine, pick a fermented palm wine because it's having the organism, maybe invertase or invertase enzyme or stuff like that. Put it on fermented one. You have introduced organisms into the new palm wine, which before you know it, it will ferment it. Production of fufu is fermentation. Use enzyme, genetic engineering. Production of yogurt is by enzyme, be lactase. So when you have a corn already fermented yogurt, pick a little and put it inside freshly produced milk. The fermentation bacteria will ferment your new milk. Manipulation of organism, production of wine, production of fufu, production of. Tell me, fufu. You know what is fufu? If you do, thumbs up. If you do, thumbs up, fantastic. So genetic engineering, production of palm wine, fermentation of palm wine. Even in our building, production of wine, production of beer is genetic engineering. Because we are manipulating organisms. So this even production of brukutu and local beer is genetic engineering. So it is not really new thing, but just that now we are doing it at molecular level. Before we are doing it at organismal level, where the organism will carry the organism and use it to manipulate our environment. Or we'll use cross-breeding system or outbreeding mating systems to transfer the gene. That is traditional genetic engineering done at organismal level. But now we are talking about genetic engineering at modern genetic engineering at molecular level. So when we know hydrocarbonoclassic bacteria, which are known to feed on hydrocarbons, and by so doing, they are cleaning the environment for us. And we don't have large number of hydrocarbonoclassic, but we have large number of E. coli, we have large number of staphylococcus. So we can isolate the catechol gene that is conferring the ability to feed on hydrocarbon in hydrocarbon bacteria, isolate them, insert them into other bacteria. They become transgenic bacteria carrying catechol gene, which we're able to clean our environment because they are carrying catechol gene that is responsible for I want a compatibility on them to be able to feed on hydrocarbons. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. Now the question is, how do we transfer gene at the molecular level? You have known how we can transfer gene at organismal level using outbreeding methods, mating systems, mating methods. But how do we create transgenic organisms at molecular level? Meaning that we are talking about gene. How do we do it? Then we talk about transgenic techniques. Transgenic techniques are techniques we use in creating transgenic organisms. What are the examples of transgenic techniques? SCNT is an example of transgenic techniques. Somatic cell nuclear transfer. Cloning is an example of transgenic techniques. Clone is an example of transgenic techniques. Cloning, you use, what is cloning? Laboratory-based manipulation of identical copies of millions of copies of an organism. Laboratory-based manipulation of identical copies of an organism, so that organism will have identical genetic makeup between the parent and the daughters. That is cloning. That is a layman's cloning. To clone it, to create an identical copy of something. So when you create millions of copies of that, you are doing cloning. When you create a genetically identical copy of something, an organism of a cell, is cloning. Am I speaking sense? Am I speaking sense? Am I speaking sense? So for us to be able to do genetic engineering, I told you, SCNT, somatic cell nuclear transfer, is an example of transgenic technique. Cloning is an example of transgenic techniques. Now, for us to be able to perform, to produce transgenic organisms, we need transgenic techniques, we need gene transfer method. Gene transfer method is called transfection. Don't forget, genetic engineering is a form of recombinant DNA technology. Transgenic technique is a form of technology. Transgenic organisms are carrying recombinant organisms, recombinant DNA, because they are carrying DNA from two different organisms. That's why we call them transgenic organisms, produced via genetic engineering concept, where we use recombinant DNA technology. So recombinant DNA technology, it can also be called transgenic techniques. Cloning is one. Somatic cell nuclear transfer is one. I'm going to take time to explore this cloning. Now, what is somatic cell nuclear transfer? And let me explain. But before I explain that, let me explain briefly. Like when there was a time in Roslin Institute in England where a transgenic sheep was produced. A transgenic sheep carrying human mammary gland gene. A transgenic technique that can produce transgenic animal sheep that can produce human milk, meaning that it is carrying human mammary gland gene, the gene that can produce human milk. So they call it Dolly sheep. Dolly sheep was created in Roslin Institute. Dolly sheep was created in Roslin Institute in England. Dolly sheep is a transgenic organism. Is a transgenic sheep. Is a transgenic sheep that is producing human milk. And you're going to wonder how can sheep be producing human milk with the aid of transgenic techniques? with the aid of recombinant technology. It is doable. It is just at organismal level. It cannot be done, but it can be. What cannot be done at organismal level? What cannot be done at the level of traditional genetic engineering, but a whole lot of it can be done in. It can be done in. A whole lot of it can be done using molecular tool, using modern genetic engineering. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking? Can you hear me? Hello? If you're hearing me, thumbs up. 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 Hello? Fantastic. Fantastic. Okay. Network is misbehaving here, but we'll not give in. We'll not give in. We'll not allow network to frustrate us. But like I was saying, so we have dollyship. I'm trying to explain. Now, I've told you, genetic engineering uses recombinant DNA technology, which is also regarded as transgenic techniques. Am I speaking sense? 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 Fantastic. Fantastic. So, in 1997, in Roslin Institute, a transgenic somatic cell transfer, a very unique and fantastic and a game changer transgenic technique, which is also regarded as recombinant DNA technology, was used to generate dollyship that was able to produce human milk. Meaning they were able to, at molecular level, transfer mammary gland gene responsible for production of human milk, was able to that is integration, to transfer it into the sheep. And the sheep was able to produce human milk. The reason is this, that there is a disease called maybe hemophilia, hemolysis is, I can't remember the disease called, is it hemolysis, inability of blood to clot. Inability of blood to clot. So, and what to use to attend to fix that problem is in human milk. That is inability of blood to clot is called not hemolysis, hemorrhage. Hemorrhage is not hemorrhage. Am I speaking sense? Am I speaking sense? Hello? Hello? inability of blood to clot. So that if you have injury, you instead of the blood to stop bleeding, it will stop bleeding. It won't stop bleeding. Some can call it coagulopathy or hemolysis. Or hemolysis or coagulopathy, inability of blood to clot. Am I speaking sense? Am I speaking sense? So, and what to use to fix this is in human milk. And to get this will be difficult. So the idea is, let us generate organism that can help us to produce human milk. The idea of dolly came. So they transfer the gene at Roslin Institute in United Kingdom. They transfer the gene responsible for production of human milk into the sheep. And the sheep was able to produce human milk because they wanted to feel coagulopathy, inability of blood to clot. That's uncontrolled bleeding. Am I speaking sense? And that is called coagulopathy. Am I speaking sense? 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 Now, there are for you to do recombinant DNA technology or transgenic, to use transgenic, you need to know what is called gene transfer method. You need gene transfer method to perform transgenic techniques or recombinant DNA technology. Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. And what are the gene transfer methods? Gene transfer method. What it is called? Gene transfer is called transfection. Transfection is the process of transferring gene from one organ to another. So we have two types of category of gene transfer method. We have transfection and transduction. Transfection involves use of bacteria to transfer gene, while transduction involve the use of virus to transfer gene. Am I speaking sense? Am I speaking sense? When the medium of gene transfer is bacteria, it's called transfection. When the medium of gene transfer is involved bacteria, it's called transduction. Am I speaking sense? When you involve, maybe if the means of gene transfer involve viral vector, retrovirus, or is involved the use of bacterial vector, like a plasmid or bacterial artificial chromosome. So we have two categories of gene transfer methods, depending on in the medium of gene transfer. Now let us delve into gene transfer method. We have vector method. We have plasmid method. We have plasmid method. We have liquefaction. We have electroporation. We have microinjection. We have macroprojectile bombardment. We have what? Micro projectile bombardment. we have micro projectile bombardment. Holistic method is also called micro projectile bombardment. We have electroporation, we have micro injection, we have gene knocking and gene knockout method. And so in the next class, we'll now begin to dwell in individual methods. We'll dwell on individual methods. Am I speaking, sis? 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 Fantastic. So you have seen category of gene transfer, transduction and translation based on medium of gene transfer. I'm asking to explain, discuss gene transfer method, the two types of gene transfer method based on, or two forms, or let me say two forms, or two categories of gene transfer based on medium of gene transfer, transduction and transfection. The technology of transgenic techniques, you must have a good knowledge of gene transfer method. How do we transfer gene? And we have medium of gene transfer. You can transfer one, we have plasmid method. Two, we have, and what is plasmid? When you are using plasmid as a vector, let me say vector method or plasmid method, but it's saying similar same thing. Vector method is involved, vector method, example plasmid. Vector method involves use of vector as a carrier of gene, as a medium of gene transfer. Am I speaking sense? Am I speaking sense? Am I speaking sense? When you're using vector as a medium, as a carrier, the medium of gene transfer. And what is the example is we have two types of vector. We have bacterial vector and viral vector. Because it's carrier. Plasmid, for instance. Plasmid is an example of bacterial method. Plasmid is a circular DNA of bacteria. The genetic material of DNA, of genetic material of bacteria is found in plasmid. The circular DNA of bacteria is called plasmid. So we can use plasmid to transfer gene. For instance, if you want to transfer catechol gene, in fact, let me give another example. I'm going to give you two examples. You know, now in advanced countries, when you're diabetics, just go to hospital, they'll do gene therapy for you. You just transfer, you inject insulin into your body system. And the diabetics has a lot of inability of insulin to control blood sugar level, isn't it? Isn't it? Inability of blood of insulin to control blood sugar level. And what aggravates, what leads to diabetes. So we have isolated diabetes, the insulin gene, and we can do cloning of insulin gene to produce thousands of millions of copies of insulin that we can use as gene therapy for people having diabetics. Am I speaking sense? Am I speaking sense? Fantastic. Fantastic. So we use cloning. In cloning, you need gene transfer methods, which is vector method. Which is what? Vector method. You can use them. An example of vector method is bacteria. But example of vector method is plasmid method. Now, how do we do it? I'm going to speak to that extensively under cloning. But let me briefly speak to it. When we will isolate plasmid from our bacteria, we will cut the plasmid. This plasmid at restriction site. We will cut the plasmid with restriction enzyme. Like, for example, EcoR1 at a restriction site, it will open. When this plasmid open, it open with stagger cuts. It open with stagger cut, means zigzag end. So we will now introduce, all of this will happen inside test tube. We'll now introduce our insulin gene into it. We are using plasmid, but black plasmid, which the carrier, the vector method. Now, that called enzyme ligase will be introduced to seal the staggered end together. So immediately the insulin gene is inserted into the plasmid. It is being sealed with enzyme ligase by process of ligation. Then we have created recombinant plasmid. The plasmid carrying foreign DNA is called recombinant plasmid. So this recombinant plasmid will now be transferred into a host organism, a host cell. A host that can be virus, a host that can be bacteria. Am I speaking sense? Am I speaking sense? So now, for instance, we are using bacteria, E. coli. So we put the recombinant plasmid inside the test tube, E. coli inside the test tube. We have different methods of gene transfer again that we can use, whether electroporation, whether, for instance, in this case, we'll use electroporation, or another method, another one is a sodium chloride solution. But in this case, we use electroporation. So that what we call electroporator. The test tube will be put inside electroporator with two terminal anode and cathode. It will create electric shock. What it does is to create electric impulse on the cell membrane so that the holes of the bacteria will open, and via osmosis, movement from region of higher concentration. No, osmosis, movement from region of higher concentration to that of lower concentration by osmotic pressure. Osmotic pressure, in biology, we're taught osmosis, movement from region of higher concentration to that of lower concentration. So when the electroporation is creating an electric impulse, it is vibrating and causing, it would cause the cell membrane to open, the pores of the cell membrane to open. Through osmotic pressure, the recombinant plasmid will enter inside the host cell, E. coli. But not all the cell will imbibe, will take up all those cells, all those recombinant plasmid. But larger chunk will take it up. Then it's now left for us to check which bacteria take up recombinant plasmid and do not. So we pack all of them inside penicillin medium. Inside penicillin medium, the transgenic bacteria will generate penicillin resistant gene that will make them to survive inside penicillin medium. Those ones that are not transgenic, that did not take up the recombinant plasmid will die. They were able to eliminate those that didn't take up the recombinant plasmid, and we are able to have those that took up recombinant plasmid. So the bacteria that take up the recombinant plasmid is called transgenic bacteria. We have created transgenic bacteria. Am I speaking sense? In that one, I've invariably discussed cloning. We must play. So now the cloning release begins. You know, bacteria reproduce by binary form of reproduction, binary fission. They enjoy asexual form of reproduction. Asexual form of reproduction. And the cell is by binary fission, the recombinant plasmid is multiplying. So at the end of the day, we generate millions of copies of recombinant plasmid, transgenic bacteria carrying recombinant plasmid carrying insulin gene. Then we put all of this together and form a gene library. Anytime we need gene therapy to transfer insulin to an individual that needs insulin gene, we just take the transgenic bacteria, isolate the insulin gene from it, and transfer. We can transfer through maybe the skin, through the muscle. We can do injection through the muscular method, or through the subcutaneous. It works faster, and the manifestation of that gene is faster if used through the skin than maybe through the muscle. You can take one week to 2 weeks before the cellular system will take the foreign DNA and before they begin to manifest itself. But if you take through the subcutaneous to the skin, within hours, it will take up and manifestation will begin to happen. Applications. So that is cloning. In cloning, we speak to recombinant use of vector method. We speak to electroporation. At times you may not use electroporation. You can use sodium chloride solution. Where you will have salt and water solution. Salt and water solution. That is sodium chloride solution. About 42 degrees. So we can use sodium chloride solution to can create a means for osmotic pressure to happen. You can use that too. Or temperature. I use temperature too. So that's how you can modify gene transfer method. So I've spoken to vector method. So, but let me speak further. I've spoken to plasmid method, the circular DNA of bacteria that we use as a vector, as a carrier for gene transfer. So let me now speak to vector. We have bacterial vector and viral vector. Example of bacterial vector is plasmid, which I've spoken to. We have bacterial artificial chromosome. Bacterial artificial chromosome is a form of viral vector, microbial vector, or bacterial vector, or bacterial vector. We have bacteriophage, the combination of phage and bacteria, phage and virus. So, but majorly, we have bacterial vector and viral vector. Example of bacterial vector is plasmid. Example of bacterial vector is retrovirus. We have phage, the coated end of phage. That is called sequence. The coated end of phage and plasmid. It gives us that they are genetically engineered to give us cosmate. Bacteriophage is genetically engineered. When you when virus attacks bacteria, it creates an organism called bacteriophage. So we can use bacteriophage as a form of vector. It's genetically engineered. Bacteria artificial chromosome 2 is genetically engineered. Yeast artificial chromosome 2 is genetically engineered. All these are vectors. Cosmate is a vector. Combination of cohesive end of phage and plasmid is a vector. These are different types of vector that we can use in gene transfer. And the retrovirus uses RNA as a genetic material. You know, some larger chunk of virus use RNA as genetic material, but we have some virus that have DNA, like adenovirus, virus that cause flu, has DNA as a genetic material. So, and retrovirus use RNA as genetic material. Upon infection, it comes back. See, let me tell you how the retrovirus causes HIV. It does what? It uses its RNA to generate complementary DNA. No, complementary DNA. To be able to generate complementary genome. So when it uses the DNA. No, it uses the DNA of, it's carrying RNA. So it uses the DNA of the host to generate a DNA copy from its RNA sequence, so that you're able to integrate itself. And as the organism is multiplying its genome, the retrovirus is multiplying too. That is the technology. Am I speaking sense? 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 So, am I speaking sense? Fantastic. So, I've explained what are vectors. I've explained vectors. So, we have. If I can give me example of viral vector, give me example of plasmid bacteria vector. I can say, give me example of genetically engineered vector. Yak genetically engineered, bacteriophage genetically engineered, can be genetically engineered, and bacterial artificial chromosome can be genetically engineered. Am I speaking sense? Am I speaking sense? So another form of gene transfer, we have micro biolistic or microprojectile bombardment. For this method is principally used for plant. When we have something like a bead, it involves use of gun-like structure. And we have a naked, something like a bead. So we use this bead. We will infuse our DNA around this bead to get a kind of a naked DNA. So this naked DNA will be coated on this bead, and this bead will now be inserted into that gun-like structure, and it will use it to shoot it into the cytoplasm of the plant. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? So it's a traditional method. It's a crude method. It's labor intensive, it's laborious, but it's very effective. Method of gene delivery, gene transfer. When you use biolistic method, or what I call macroprojectile bombardment, it delivers the DNA into the cytoplasm. Majorly, all these genes are, when you introduce them into the cytoplasm or into the nucleus, they're more or less extra chromosomal DNA. They more or less behave like extra chromosomal DNA, extraterrestrial DNA. They don't really, some of them, they don't really commingle with the genome of the organism. But the retrovirus will do. Retrovirus will do. But this, like, for example, like this biolistic method will not commingle itself to the DNA of the genome, but it behaves as extraterrestrial, extrachromosomal DNA, in the manner that when it shoots into the system, as the cellular system is growing, the DNA will manifest its character. And that is why, since they cannot commingle itself to the genome, to the DNA sequence, then the F1 cannot reproduce itself. So in this one, they should do it is effective. For instance, that we use in rice, that we use in rice, in corn, and in several rice, corn, several cereals, several grains. It has been used. So we use this one to create genetically modified organisms, genetically modified crops, like BT coin, I mean, like BT cotton. We have now beans. So now certain variety of beans have been produced that have thick tester that Calotobacter maculatus cannot penetrate. We have the varieties of beans. Some are hard. That, for instance, we have local beans. There is one local bean that is common in Southwest. It's common in Southwest, the local breed of beans. You have thick tester, thick back. That will take a longer time to cook. When you cook it, the water will be red. Bean weevil always find it difficult to penetrate such hardcore tester. You can express the gene that make tester to be thick. Insert it in our brown beans and all that. And we have a very bean that has a thick back that Calotobacter maculatus, bean weevil, cannot penetrate. So in that kind of research, or kind of research adventure, you use biolistic method or what we call microprojectile bombardment. So for instance, there are some cultivators that is going, that those that are producing GMO crops, want to impoverish Africa. They take sole ownership of the seed, that the seed cannot reproduce themselves. That they want us to plunder or to destroy our local genetic resources, our local seeds, and now begin to buy from them. A point will come, our local seed will go into extinction. Then we'll rely on the West for our seed. We rely on the West for our food. That is a archaic form of mentality. That is ignorance to the highest level. The idea is simple. We need to rely on the West for genetically modified crops. We can generate our own. We can have because the patency of genetically modified crops, nobody can have it except the owner. We can generate our own and maintain the patency. The reason is this: when you use biolistic method to deliver gene to the cytoplasm of a plant. And like I told you, such a DNA is extraterrestrial. It's not commingling into the DNA of that plant because it's in the cytoplasm. As the cellular system is growing, the plant will manifest the characteristics of the gene you have inserted. So the F1 you generate cannot reproduce. If it does reproduce, it cannot produce itself because that gene you deliver is extraterrestrial, it's extra-chromosomal. That is why we need to produce F1, 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 F1. And if you want to reproduce F2, okay, you have planted the one you bought from Europe or from seed company, you planted it. When you plant it, to give you the heterosis, it will manifest the gene you have inserted. But the seed of that one cannot manifest the characteristics of the parent because the gene that produces them is extraterrestrial. And don't forget, another thing is this: when you begin to breed within breed, the inbreeding will come in. As you begin to use the same thing over and over again, there will be inbreeding. Homozygosity will increase. Uniformity will increase. Production performance will reduce. That's why you need to go back to the source of the seed to buy another one. Am I speaking sense? Am I speaking sense? Am I speaking sense? So we may not really rely on the West or Europe for our own genetically modified seed. We can generate our own. Technology is there. So that is what happened. That's what I'm picking to in biolistic method. Another one is microprojectile injection. And like I said, the gene required are inserted into the gun-like structure and to be inserted into the cytoplasm. Another one is microinjection method. This is when you are delivering gene to the nucleus of the cell. When you deliver the gene to the nucleus of the cell, it will commingle DNA of the cell, or it can act as extraterrestrial. When it has commingled to the DNA of the cell, of the organism, when that the F1 can reproduce itself. But if the gene is acting as extraterrestrial, extra-chromosomal, it will not. The F1 won't be able to reproduce the characteristics of the parent. Am I speaking sense? Am I speaking sense? Am I speaking sense? Now, microinjection involves transfer of DNA into the nucleus of the anchored cell. The DNA is injected directly, and it will move into the nucleus. That's the way this microinjection technology works. But the process is more laborious. It's more labor intensive than the microprojectile bombardment. But this is how we generate transgenic organisms. But here, like I said, is not really efficient. No, you can generate. It's labor intensive, but it's not really efficient. But you can do copies of about maybe 20. You have 5 that will be able to produce transgenic animal 5. So that is efficiency. But it does produce transgenic organisms. The efficiency is low. The efficiency of transgenic is low. Am I speaking sense? For example, the percentage of embryo that will undergo microprojectile injection to each transgenic organisms are low, but it will still do. Let me tell you, the roast. I'm not going to speak on dolly sheep when I'll be there dwelling on cloning. You know that about 400 out of 400 transgenic sheep that was produced, only one survived. Only one survived. It is labor intensive, it is capital intensive. Only one survives. And that one cannot reproduce itself. So you have to go and reproduce another. It is sterile. But we have to generate another one. That's how labor intensive it is. That's how capital intensive it is. So you need a lot of money to do that kind of research. Am I speaking sense? Am I speaking sense? Am I speaking sense? And like I said, microinjection is labor intensive. You do hundreds of copies of microinjection. Only few of them will take over the gene. Only two will take over the gene. Only two of them will take over the gene. And they don't usually transfer their genetic material to the offsprings. They don't. That is deficiency. But they can still produce their DNA organisms to our satisfaction. As I've spoken about electroporation, we have liposuction. Liposuction is used when you are delivering gene with use of nucleic material. They're called lipo for lipids. When you are delivering nucleic material with use of lipid organisms, a lipid reagent. You deliver nucleic acid with use of liposome, a lipid-based reagent, or a lipid-based compound like cholesterol. Like cholesterol, there's some liposome made from cholesterol. There's some lipid-based compound made from cholesterol. We can use it to cut our DNA to deliver our DNA into the whole something. Liposome is a small artificial version of a spherical, let me say, of a spherical liposome, let me say, is an artificial cholesterol. Let me put it that way. Let me just put it that way. That we can use to deliver DNA into those organisms. And it's especially useful in drug delivery into anchor organisms. So when you are using use of lipid material in delivery in drugs or nucleic material, which is DNA, into anchor organisms, that is liposuction. That is liposuction. That is liposuction. So example of example of lipid materials that can be phospholipids, can be cholesterol. Hello? Hello? Network is bad. My network disappears. My network disappears. But it's still back now. If they're hearing me, thumbs up. My network disappears. It's just coming back now. Hello? Okay, you guys are back. My network disappears. Hello? Are you back? Fantastic, fantastic. So I will be ending. I'll be calling it a day here. I'll be calling it a day here. So the next next hello? Yeah. I have discriminated between transduction and transfection. I have spoken to vet plasmid method. I have spoken to biolistic or microprojectile method. I have spoken to microinjection method. I have spoken to electro. I'm going to dwell extensively on SCNT. Somatic cell with a form of recombinant DNA technology. So next class will be dwelling on SCNT and cloning. I'll give some pictorial talk for that to further brighten your understanding. The network has not really, but we so the next class maybe by 11 o'clock tomorrow we'll start again. Let me say 11 o'clock. Should we make it 1 o'clock? If it's 1 o'clock, thumbs up. If you prepare 1 o'clock, let us thumbs up. If you prefer 1 o'clock, thumbs up. If it's 1 o'clock class, thumbs up. Mr. Sadem, Mrs. Sadebo, if you prefer 1 o'clock, please thumbs up. Thumbs up. 1 o'clock will be better. Fantastic. So I'm going to generate the video version of this class and the transcript of this class. I'm going to forward to you. Thank you for your time. Thank you for your resilience. God bless you.
This is a big advancement.